Good morning, everybody. I am glad to talk to you today about label-free quantitation. I think that a lot of people assume that if it's in the mass spec, it's got to be quantified in some way. The reality, as we're going to get into in the course of this lecture, is that mass spectrometers are very, very good at making mass measurements, or mass-to-charge measurements more precisely, but that there's quite a lot of play that comes out in the intensity that we measure from mass spectrometers. And over time, there have been a lot of different methods put forward about how we should go about quantifying through mass spectrometry. In general, the number of people who send a sample to a proteomics core facility for a simple inventory, what proteins are here, is relatively small. In fact, most people are trying to make some sort of question about differential proteomics. In other words, how do these samples differ from those samples? All clear? Okay, so we're going to talk then about differential proteins, proteins that change in the context of a broader proteome. We're going to talk about two key techniques in label-free quantitation: spectral counting and extracted ion chromatograms. So following on Tuesday's lecture, we're going to continue with segments D, E, and F for this session. So first off, why do we call it label-free quantitation, or LFQ? This segment is largely about all the stuff we're leaving out of the talk, because label-based quantitation has been commonplace in proteomics for a long time, and it's only relatively recently that most people decided that you could do viable quantitation using no isotopic labeling. So what are these isotopically enriched labels? The idea here is that on the mass-to-charge axis, if you have a light and a heavy version of the same peptide, they should appear pretty close together, right? And if, depending on how you've labeled them to make one heavy and the other not, will determine this gap that appears between them. Okay, so if you have the same peptide that just differs in how many neutrons it's carrying in effect, we expect that the retention time will be identical between the heavy and light, but then there will be a mass difference, which means that we have two peaks representing the same chemistry apart from the neutrons, and we can take a ratio of those heights. All fair? All right. So metabolic labeling is one of the earliest approaches that we have seen used in this, and it really relies on the fact that you're growing cells of your samples, and these cells can be grown in a way that ensures that every nitrogen is heavy, or that every carbon is heavy, whatever you're doing. So you need to have some way of metabolically labeling these creatures so that they do that. So I think most of us would accept that for many species it's possible to do this sort of total labeling, but you wouldn't really want to try it in a eukaryote, would you? It's a little more challenging to think about heavy labeling a rat, for example. That said, it has been done. It is painful to try to convince an animal to eat only heavy labeled algae, for example, but yes, an animal that's hungry enough in the lab will do that. So yes, it is possible to do metabolic labeling in this way, to entirely replace one element that's found in this organism. Of course, that's not very good for humans. I mean, I personally don't sit around and eat heavy labeled stuff all day, right? So SILAC is an approach that was put forward in large part by, really popularized by the Matthias Mann lab, but it's been used by others as well, which is the suggestion that we're not going to replace all the nitrogens in this organism, or all the carbons in this organism. We're going to instead replace all of an amino acid with a heavy variant. So if you are able to label a sample in which every lysine and every arginine is heavy, that's one way that you can force one sample to be at a higher mass than the other. But a very, very popular route is not an in vivo labeling at all. This isobaric tagging approach adds an isotopically labeled N-terminal prefix onto peptides, and we can use this to multiplex many samples in the same experiment. So a single LC-MS experiment will have 4 or 8 or even 20 different samples all multiplexed together, each with a different variant of this isobaric tag. So there's a long history of labeling our samples with heavy isotopes in order to make it possible to compare the intensity attributable to one sample to the intensity attributable to another. Okay, so this is labeled quantitation. Now, you might have an experiment that really has many samples and you want to compare fairly among all of them. So it might be that you make a mix of all of these samples, a pooled sample, in other words, and then you can label just that pool and compare each of the like samples to that pool. So if I know that I have the same internal reference pool in 2 different raw files, one representing the white sample and one representing the black sample, I can compute the ratio of white to internal label, to the pool, or black to the internal pool, and from that learn what the ratio of expression between white and black is in this case. That's a fractional change. So you can see that even if you have far more than 20 samples that you want to compare among, so long as you have a pooled sample carrying a label, you can make use the pooled labeled sample as a point of reference for computing all of those. Okay. So why would we do away with this? Why would we push that to the side and say, this is not the way we're going to do things? Well, first off, heavy reagents cost money. And I think everyone knows that research budgets are always straining to cover personnel and equipment and services and so on, and reagents can add quite a lot. So rather than pursuing, say, the iTrack route or the SciLac route, someone might try to do quantitation that puts one sample in each LC-MS experiment and makes comparisons across experiments instead. So now we don't have the ability to compare within a single raw file, within a single LC-MS experiment. We need to compare data across LC-MS experiments to compare samples. In some cases, this is quite necessary because a pooled sample that represents all of the samples is not really feasible. What if you're receiving one batch of the samples in February and another batch of the samples in June? Are you going to run the data in such a way that you can only compare among the Junes and among the Januaries? It's kind of a mess, right? So a situation where you have to wait until all the samples are available before you make a pool can actually delay the start of a major quantitative experiment by months. And certainly incomplete labeling is a very big deal. What if only 90% of the lysines in a sample have been heavy labeled? That's going to create all kinds of havoc with the relative enrichments. The heavy sample by itself would already appear to have some light sample in it. So that's kind of a mess. So these have led to people really emphasizing label-free quantitation as the means for comparing samples via proteomics. I would note that although label-free quantitation has been around for quite a while, the phrase label-free quantitation really started showing up around 2003. So people have been doing spectral counting and intensity assessment through extracted ion chromatograms for a long time. But this term, the moniker label-free quant, is actually a fairly late addition. Where before, I might have a single LC-MS experiment that represents multiple samples pooled together or combined in a way that they can only be differentiated by their isotopes. We change to a place where each LC-MS experiment is about a single sample, which means we must compare across LC-MSs. Label-dependent techniques are more expensive, and there's also a widespread perception that label-dependent methods, these strategies using iTrack or SciLac or whatever, also produce more reliable quantitation. I'm not sure that really holds water. Having performed differential proteomes in data sets that involve the same samples, but some were done by label-free techniques and some were done by iTrack, it's not entirely clear to me that the perception that label-based methods produce more accurate quantitation is actually true. So I would consider that one as an unyet proven assertion by the folks who rely on label-based quantitation. So the stars of the show today are going to be spectral counting and extracted ion chromatograms. Spectral counting and extracted ion chromatograms. We're going to start with spectral counting because I think it's probably one of the simplest things to explain. But honestly, although I describe it as simple, I realize that a lot of people in the community have a lot of misperceptions about it, so I'm going to try to walk through it relatively slowly. Let's start with the underlying hypothesis that if you have more of a protein in a common background, rather than less of a protein in this common background, the number of peptides we can see from it is larger when you have more of the protein than when you have less of the protein. That may not be obvious, but I would try to put forward a model where we think of each protein as an iceberg. And if you have steady seas and there's no ice around, you can see plenty of that iceberg. But as soon as you're in a really messy background with all kinds of icebergs floating around, your ability to see deep into that iceberg is abolished. That's maybe not a very good metaphor, but we'll work with it. Each protein has high probability peptides that you're almost certain to see, and some peptides that are much lower probability. Let's say you've got a missed cleavage peptide, and it only forms if the middle cleavage point isn't cut, right? So that would cut into the probability that we're going to see it. What if you have a peptide that shows up at a spot in chromatography where lots of peptides are eluting at the same time? There's a lot of competition, not just for protons, but for that peptide ion to be selected for MSMS. That's a low probability peptide, right? So for any protein, there's going to be stuff that necessarily shows up if this protein is there, and some stuff that's going to show up at a very low probability. As a result, when there's a lot of a protein present in a sample compared to very small amounts of that protein in a sample, we expect to see more peptides from it. It is also the case that a peptide at high concentration can also show us multiple looks when it's at high concentration. So a peptide ion may show up in multiple charge states. For example, it might show up as a +2 and also as a +3. It might just have a very wide peak in chromatography if there's a lot of it. As a result, you take multiple shots at it in tandem mass spectrometry. It might be that it has PTM variation, that let's say 1 in 10 copies of this peptide are phosphorylated. If there's a lot of that protein there, you might see it. But if there's not very much of that protein there, you might not notice the 1/10 of all copies of this peptide that are phosphorylated. So there are lots of reasons why a peptide can also show up in multiple spectra rather than just one in the experiment. So we can compare the number of PSMs matching peptides from a particular protein among LC-MS experiments. So remember that for a given protein, we have multiple peptides that might be seen. For an individual peptide, we may have multiple PSMs that come out of it. We're going to aggregate all of that together at the level of protein. So if we had 5 spectra from this peptide and only 1 spectrum from this peptide, we're going to sum all of those together and say, for this protein, we have this many peptides and this many PSMs. Fair enough? Okay, so that's the most common way that we quantify how much of a protein is present in spectral counting. So why does it work? The mass spectrometer has limited attention available. It can generate, let's say, 10 tandem mass spectra every second. That's really fast compared to the stuff that happened when I was in grad school. But you can capture 10 tandem mass spectra in a second with, say, a contemporary Orbitrap instrument. So the instrument is trying to select, for fragmentation, those ions that are most promising, which is to say they have the greatest intensity, suggesting there's a lot of them there, and it's an ion that isn't at a mass to charge value that was recently sampled. Okay? So that creates a sampling process. I kind of think of a mother bird coming back to the nest with a nice big worm, and all of the chicks are all squawking at her to, I want the worm, I want the worm, I want the worm, right? And the one that squawks loudest gets the worm. So if you've got a not thriving chick it's going to become less and less able to compete for the worm brought back to the nest because its brothers and sisters are all chirping too loudly. Okay, in the same way, a peptide ion that is of middling intensity, not a huge one, that's in a really crowded time of the LC-MS landscape, may not be sampled for tandem mass spectrometry consistently. This is a competitive process. Okay, certainly different trypsin cutting sites on a protein get chopped at different probabilities. Yes, they're both lysines, but one lysine is embedded in a section that's kind of resistant to denaturation, and the other one is in a spot that's a nice straight chain ready to go. These are different probabilities then of trypsin cutting. The peptides must compete for protons. There's this thing called ion suppression, that if you have a whole bunch of peptide ions that are all trying to ionize at the same time, there's just too few protons to go around. Some of them don't ionize to begin with. And certainly, you may produce an MS-MS scan that fails identification too. It could be that it's too low a signal to noise. It could be that it is a spectrum that results from two peptides that have co-eluted, that come off the column at the same time and have very similar mass to charge values. There are lots of reasons why an MS-MS may fail to be identified. So what comes out is usually a table that looks something like this. These are, by the way, real data. This is an experiment we did back in 2009 on an Orbitrap Velos. We had 5 experiments that represented red E. coli, and we had 5 experiments that were yellow E. coli. We're just calling them red and yellow because those were the colors we outlined them with on our gel in the end. But we know that a subset of these E. coli proteomes are differential because we cut those proteins literally out of the gel. So we know that they're gone, mostly. All right, so now we have these numbers of spectra, numbers of PSMs, that relate to peptides from this protein in replicate 1 of red, in replicate 2 of red, in replicate 3 of red, and so on. Now, I know a lot of people in biotechnology are very fond of 3-on-3 experiments. You do 3 replicates of red and 3 replicates of yellow. One of the things that I really, really appreciate about our most common collaborator in mass spectrometry here is that they've really pushed people to start doing 5 replicates rather than 3. And it leads to a better ability to discern which proteins are differential between one cohort and the other. Now, I note that PowerPoint is only letting me show this many rows, but there are actually something like 500 or 600 different proteins that we detected well here. So we've got 500 or 600 rows of this stuff. How do we go about creating a metric that tells us which protein differs between these 2 cohorts? I would think the first answer that comes to most people's mind would be something like Ttest. But in fact, there are some reasons why you don't want to use Ttest on this. We'll come back to this in the last segment of today, okay? Now, all this is quite different than the extracted ion chromatogram. Have you seen a mass spectrum in some of our previous lectures? I know I showed a tandem mass spectrum on Tuesday, but have you seen some mass spectra along the way? Okay, the short version is that a mass spectrum is showing you a catalog of what ions are coming off chromatography right now, okay? Each of these mass spectra is shown as a horizontal dashed line on this plot here. So each mass spectrum is usually taken, say, a couple seconds apart from the last one, maybe 3-4 seconds from the last one, depending on how you've configured the instrument. But if you have a peptide that elutes over the space of about 30 seconds in the way we've configured our columns, you might expect then that if you're measuring a mass spectrum every 3 seconds, that you get 10 shots on it. You get 10 snapshots of how intense it is at this moment in time. So, if you were to look at the same mass to charge value across this narrow range of times, across these 30 seconds, you're going to have some measurement of a peak associated with that particular peptide ion. This is assuming that you can distinguish this peptide's ion from all the others by mass to charge. If you're doing this in an ion trap, forget it, it's going to be a mess. But in say, FT style, Orbitrap style instruments, you have really good resolving power. And so ions may be very close together in mass to charge space, and you can still tell them apart on each mass spectrum. So here we have kind of a good shape for what a peptide ion usually appears to be. You have some inkling that the ion is just starting to appear, and then it suddenly explodes up to its maximum height, and then it falls off rather more slowly. Some people have described this as a log normal shape. So rather than being symmetric like a bell curve, it's usually kind of pushed to a side. So this yellow one is showing us the kind of shape we would expect, a rapid rise and then a fall off again. The red one, a nice rise and fall off again. But look at this blue one. It's a little funky, isn't it? We've got a rise and a fall off, and then it re-rises and falls off, and they're both colored blue. Now, are these in fact the same peptide? Could they be two different peptides that show up right after each other in retention time at very similar mass to charge values? Could be in some cases. So this could be what we call a split peak, that something has caused this peak to be separated into two humps by something that happens, maybe a sputtering source or something like that. Or this could be two different ions that coincidentally are at consecutive retention times and at very similar mass to charge values. Okay? So an XIC, an extracted ion chromatogram, is one of these colored shapes representing some particular peptide across some short range of retention time. Most of our experiments are like 90 minutes long, so a 30-second period within this is what we're zooming in on. We're looking at just a tiny slice of retention time and just a tiny slice of mass to charge in this diagram, but it's useful for understanding this. What's the other thing that would show up here? Well, we would expect that we don't just have one ion representing each peptide. You would actually expect that each peptide shows up in different isotopic forms, right? So you have, say, 1% of all carbons are C13 rather than C12. As a result, you expect to see a pretty substantial amount of intensity showing up spaced out as isotopes in different XICs. So if you're trying to integrate the signal for this entire chromatogram, you'd really like to have all the different isotopes for it as well. Okay, so we're going to do this process of integration in order to translate each XIC into a peak area. That area is our clue to how much this peptide's concentration contributes in this experiment. All right, now there's going to be a bit of a disconnect as I move forward because I talk about XICs as relating to peptides, but in the end we want to differentiate proteins. And the way that we turn peptide XIC areas into protein areas is frankly a little muddled. Everyone seems to have a slightly different way to do it. So I'm probably going to just skip over a bit of that and just say we have some magic that lets us transmute peptide areas to protein areas, okay? So is it only possible to generate XICs from peptide ions? And the answer is no. There are different ways you can configure the mass spectrometer, such as selected reaction monitoring or data independent acquisition. I've put them down at the bottom here. These methods allow us to quantify not just on the basis of the intact peptide ion, but on the basis of the fragments they produce. So we're producing chromatograms not just for precursors, we're also looking at the fragments that come from them. So the software Skyline is really, really good at handling data of this sort. Here you can see that in this narrow range of retention times, we have, I guess, a burgundy, a lilac, a teal, a dark blue, and an orange. 1, 2, 3, 4, 5. All 5 of these Y ions associated with this particular peptide are all rising and falling together at a common retention time. This gives us the ability to quantify this peptide much more accurately because it's possible that we would just have a rise and fall in some random part of the retention time that doesn't have anything to do with this peptide. So by playing these signals off of each other, by recognizing that all of these fragments attributable to this peptide are rising and falling together, we have a better chance of pinning this down. Okay, now, what happens if I integrate the area for a peptide that was identified in 4 of my 5 experiments, but in the 5th experiment it was not identified. What does that look like from a statistical point of view? Well, I want to compare these 5 values for the peptide to these 5 values for the peptide, and now one of them, we didn't identify it. Well, that's a problem. And that's what match between runs is all about. Match between runs was really popularized by some software called MaxQuant, published back in 2008. Match between runs gives us the ability to predict where in this experiment this peptide should have been and go integrate it even though we didn't identify a tandem mass spectrum from it. That's kind of tricky, so let's walk through it. First off, if I don't identify the peptide and I don't have a chromatogram, an XIC, to work from, I have a missing value in my table. And statistics hate missing values. If you know what it will do to your data analysis, you will also hate missing values. So we want to be able to find this intensity even though the peptide wasn't identified. So what we're going to do is expect that this peptide ion was visible in the MS of this missing value raw file, but we expect that if a tandem mass spectrum was produced, it was not identified successfully. So what we need to do is figure out what retention times we need to look at in this missing data file to say between what 2 times do we expect to see its chromatogram. Now chromatography is just a little bit slippery. If you see a peptide at 17.8 minutes here, it might be at 17.6 minutes here, or it might be at 18.3 minutes here, or it might be, you might have very regular chromatography and everything is golden. But the reality is we expect some amount of slippage back and forth. So we need some way of asking how accurately do we know where this peptide ion should be in retention time in this file. So the information we can use here is that each pair of LC-MS experiments have a lot of peptides they've identified in common. As a result, we can align the retention times between them to say 17.6 minutes in this raw file should probably be equatable to about 18.2 minutes in this one. It was just a little bit later that all the peptides appeared. Okay, so now we know the mass to charge value for where this precursor ion should have appeared, and we know the retention time range because of our alignment. And so we can go integrate that whatever peak we find there, even though no MS was identified for that peptide. It's all a little magical, isn't it? So does that always work? There is the possibility, given how hairy most MS spectra are, that even if no copies of that peptide were present in the sample, you'll still be able to integrate an intensity for it. What I'm talking about here is just integrating the noise. You're looking in a place where this peptide should have been by match between runs reckoning, but it's still possible that you get some sort of positive number from it, even if that peptide was absent. And that's what this paper by you et al. In 2021 is about. It's about an approach to estimate the false quantitation rate, effectively to say, out of 1,000 peptides we tried to go quantify despite their missing MS scans, what fraction of those do we think we've failed to integrate anything meaningful for them? It's a rather difficult problem. Okay, so match between runs is our way of patching the holes in our table to get an intensity for a peptide ion that was not identified in this particular experiment. Okay, everyone have that? Okay, it's a really important feature. So how do I decide the same raw files, the same LC-MS experiments can be used in a spectral count analysis, or they can be used in an XIC analysis? So how do I choose? Which way should I go? This is honestly a question that a lot of experienced people in our field have difficulty deciding. Largely, you'll find that people have great familiarity with a software workflow that relies on XIC, and so they assume I should use XIC. Some people really trust spectral counting, and they use that all the time. So a lot of this is going to rely on familiarity, but there are some reasons why you might opt for one rather than the other. First off, if your biomarker test, if your differentiation of two cohorts is entirely based around which proteins changed and which ones are up and which ones are down, spectral counting is quite good enough. It's perfectly good for that. Being able to determine which proteins are differential and in which direction is kind of the most fundamental thing that most people care about in cohort versus cohort examinations. It might be, though, that you have a much more quantitative question in mind, which is not just which ones are up and which ones are down, but by how much are these up? By how much are these down? Is this up 20%? Is it up 400%? I mean, if the only protein differences you detect are up by 400%, that's kind of, you're limiting your power quite a lot, right? So these questions actually matter quite a lot. It might be enough to you to know these proteins are up, in which case spectral counting is a completely fine way to go about it. Okay, next up. It might be that you're dealing with a very, very large number of samples. Maybe you've got 100 samples. Doing all of those in one big batch on the mass spec is kind of dubious, because the mass spec may not be available to you for, say, 200 consecutive hours. It might be a core facility that can give you 60 runs this month and 60 runs next month, but not all at once, right? So for something like that, it might be that you need something that is less sensitive to chromatographic variation and intensity variation. If you're going to be distributing your samples in multiple batches, spectral counting is actually more robust to that kind of messing about than is XIC-based analysis. Okay, finally, if what you're trying to evaluate is the quantity of particular peptides, for example, to ask what fraction of these peptides carry phosphorylation as opposed to being nude of phosphorylation, then doing something like that via spectral counting is a non-starter. You really would need to use XIC-based analysis to work your way through questions like that. So I hope that these are useful for you in trying to evaluate which track you want to go with the data set. I just want to point out that if you read a paper by somebody in proteomics that you really respect and you think this data set's really cool, and they did their analysis with XICs, there's nothing that prevents you from downloading those data and doing a spectral count-based analysis on it too. Most authors, as I've said, are going to use what's familiar to them, not necessarily the tool that you are most comfortable with. Okay, plenty of takeaways, but the short version is that spectral counts are aggregating information at the protein level by summing across all the PSMs for all the peptides. This means that the count that comes out is kind of just one number for the protein, but it's a relatively easily interpreted one. Among all these peptides, among all the PSMs, we saw 53 spectra that were matched to some peptide for this protein. Okay? XICs are going to compute intensity areas for each peptide. They're going to compute they'll probably compute ratios for these peptides, and from that try to give you an aggregate protein ratio built from peptide ratios. Okay? Hanging in there, right? Doing all right? Good. Excellent. On we go. So this part of the talk is a little bit longer, and I hope that I can have you following along on each step. I have included a little bit of code, so we'll try to walk through that as we go. Does anyone need to take a break at this moment? Okay, I'm going to plow ahead then. So let's start with this saying that mass spectrometers are masters of the x-axis. Mass spectrometers are really, really good at resolution, at being able to discern between ions that have very, very similar mass to charge values. And they're very mass accurate. Given a mass value from a well-calibrated mass spec, you know this particular ion's mass to charge very, very accurately. However, intensity measurement is always a lot bouncier from a mass spec. It might be that when you're looking at ions that are not the dominant one in their mass spectra, but they're the little minor contributors on the side, that if we look at their stability of intensity across their chromatograms, we see an awful lot of jaggedness. Okay, so this gives us the concern about coefficient of variation, which is to say, we've computed the area, for example, for this peptide across these 10 samples, we know that exactly the same amount of sample was injected in each case, and yet we see that we have some variation that we've observed. So a 10% CV value is what came out of this big SRM assay published several years ago. The coefficient of variation is, I mean, it sounds like a very impressive number, but it's not actually that complex to understand. Typically, we're describing the standard deviation of the measurements divided by their mean value. So if I were to compute that for this classroom, I might write down everyone's height in centimeters. I would then compute the mean for it. I would compute our standard deviation, and I would ask, how big is the standard deviation compared to the mean? So I don't know what the range of heights is here. It's what, 20 centimeters height difference? And if our actual heights are, say, 160, 20 versus 60 is a pretty small number, right? I mean, it's not a 20, it's 1/8, so that's 12.5% CV. That's about what you have in the intensity variations that come from selected reaction monitoring experiments. Okay, on we go. So, calibration curves are very, very important if you're talking about quantitation. Now remember, I've already said that in looking for proteomic differences, you may simply be asking what changed and in which direction. In a case like that, you're not really quantifying. You're just asking, did it go up or did it go down? That's hardly quantification. But it might be that you have an experiment where you need to be able to accurately quantify exactly how much this protein has risen or fallen. And for that, you really need to go the route of a quantitative experiment and construct something like this calibration curve. Here we're seeing that even though the peak ratios, the known peptide concentrations coming into this experiment have fallen and fallen and fallen, we see that we're still measuring intensity for them, even though they're not actually present in the sample at the lowest level. This is pretty common. Remember that I said if you're simply integrating what you find where you expect to find the peptide, you might find a peak even if that peptide is absent. So it might be that you get a little bit of intensity measured even when the thing is absent altogether. This tells you a lot about the limit of detection and the limits of quantitation. These are concepts that show up again and again. I've talked about them in other contexts as well, but in the case of quantitative experiments, being able to understand whether you are in a range where you could accurately quantify a given peptide is certainly a big deal. Okay, now normalization. If you see me talk in the next-gen sequencing class, I talk about normalization there. I went in search of a good paper on the role of normalization in label-free quantitation, and I found a really cool paper from 2019 on this subject. So we start with the idea that one of the things we may wish to normalize is the arrival of peptides from the column, from liquid chromatography. There are peptides you can purchase, called IRT peptides frequently, that you spike into your sample, and you know their expected hydrophobic performance. When should they show up? But by seeing when they actually show up, you can help to calibrate the liquid chromatography separation that you're using for peptides. Very helpful. Okay, so IRT peptides can help you make your retention time adjustments much more robust. You might also think about what I typically call a minimum information criterion. This is, it's a kind of an overblown term for a simple concept. You should probably consider that trying to quantify everything you identify is probably too big an ask. It's probably a little bit too messy for you to attempt to do that. A lot of the proteins you identify have very, very little evidence, very little consistency in the evidence for them. As a result, you might want to put in a criterion that says, if I didn't see peptides for this protein in at least half of my experiments, I will not try to compute a differential statistic on it. I'm not going to run a Ttest on it. So I think that being able to rule out proteins to focus on just those that have enough information for making quantitative statements about is appropriate. It is appropriate to model the variation in each peptide's intensity. It might be that if you look across across the row for a particular peptide's XIC values, that the median value is right in the middle of the clump and there's nothing that really stands out way to the left or way to the right. That's a good case. But it might be that you see some outlier values in intensity for this particular peptide that could really screw things up if they're allowed in there. So doing some normalization to try to either coerce these peptides to a more sensible distribution or eliminate this peptide from consideration is appropriate. It's frequently the case that people who are trying to roll up peptide ratios into protein ratios will focus on only the top few peptides by intensity for it. You know, if you're trying to build a protein ratio off of a peptide that's got a vanishingly small signal, it's got a correspondingly high CV value, and that noise is just going to distort the larger protein ratio by all odds. So you should think about just focusing on, say, the top 3 or the top 5 peptides by intensity, because the most intense signals should have the least noise to them. You should expect batch effects, and you should statistically model them if it's appropriate in your study. If you run a bunch of samples in January and another batch of samples in June, don't expect that those values can all just be tossed into one hopper and run together. You should probably make use of a statistical model that has a specific term for which batch these experiments come from. So I thought all of this was really good advice, and it's a useful place to start with it. I don't remember how much I emphasized MA plots in the next-gen sequencing module, but I've been placing more and more emphasis on this type of analysis. The MA plot is an attempt to determine whether there's a bias in your data. So imagine that you've got cases and controls, two cohorts, and you want to know, do proteins at low concentrations have a bias towards the cases and away from the controls? So cancer, for example, is a disease that does all kinds of crazy stuff to protein expression. And it might be that in cancer you see a whole lot of minor proteins playing up to higher concentrations than they appear in the controls. And if you don't really have a kind of a background of unmoving stuff that you're looking for differences among, that can really mess you up. So you have to be cautious of circumstances of cohort designs where one cohort is characteristically different in almost everything from a control. All right, so it could be that one of your samples, that one of your cohorts simply had a whole lot more robust intensities than did the other. And if you have a protein at very low abundance, which is to say at the left of the plot, you might find that you have a skew pushing up where the proteins are seemingly more abundant in the cases than they are in the controls. So up and down in this case is to say more present in one cohort or more present in the other. Something sitting around 0 is at a log fold change of 0, which is to say a 1-to-1 ratio between the two. So these data came from a cool paper by Laurent Gatto explaining about the tools we have in R for visualizing proteomic differences. It's a really interesting, good visualization paper. So here we've got an MA plot for the 4 versus 2 experiment. This is an MA plot for a 6 versus 2 experiment. In both cases, to the left means a very weak signal for this protein. To the right means a very strong signal for this protein. Up and down tell us how much one cohort is favored over the other. All right, so MA plots are really useful for us to understand whether we've got biases built into the data in the main. Okay? All right, so Ttest. We all know Ttest, right? Ttest is great. Ttest works everywhere, doesn't it?
Honestly, I don't remember.
Oh, all right. Okay, so in a Ttest, we have a bunch of values of A and a bunch of values of B. And, the values are the same thing, but in the A cohort and in the B cohort. And now we hypothesize there's no difference between these values from in the values we get from either cohort. We run the Ttest, and the Ttest provides a p-value that says whether our hypothesis that they're all the same holds up well, essentially. So a low p-value tells us that our model, that the values should be all the same regardless of what cohort they come from, is not a very reliable one. All right, so that's our null hypothesis, that the two distributions have the same mean. We have some test statistic, a t-value, that we compute from these actual values in the two cohorts. And now we want to know, is it probable that we would have a higher or lower t-statistic by random chance? Gosset's introduction of the t-distribution allows us to compute a probability that we would have a more extreme value. And that's a p-value that comes out of a t-test. Does everyone know where Gosset was working? You've heard this called student's t-test, yes? It's called student's t-test. Oh yeah, okay. So Gosset was this kind of interesting guy. He's a mathematician working for the Guinness beer brewery up in Dublin. And the folks at Guinness were a little hesitant to let anyone know they'd hired a mathematician, because they didn't want the other breweries to go hire mathematicians and gain the same way that they did. So they didn't let Gosset publish under his own name. Instead, when he published the t-distribution, he was forced to publish it under a pen name, and he chose the name student. So student's t-test was Gosset's test for differences. He just wasn't allowed to publish it under his own name. He was kind of an interesting guy. Anyway, I gotta say, out of all the people I teach about in biostatistics, Gosset is probably one of the people I loathe the least, because a lot of the people behind biostatistics are vile people. I won't get into that right now, but it's true. That's fine. So given a t-statistic, we can then compute a probability that we would see a more extreme value by random chance. So you've got, let's say we all go on a diet. So we write down our weights in kilograms, our masses in kilograms today, and we come back a month from today, and we all write down our masses. So are they different or are they not? That's one hypothesis we could use. We could also say, did we substantially lose kilograms by going on a diet for a month, right? So that would be a paired test, because you wouldn't want to compare, my post weight to your beginning weight. That would be nonsense, right? So t-test has a lot of different flavors that can be used for handling different scenarios. But t-test is not for everything. So I said I was going to show you some code. Here's our code. This is a case where we are using spectral counts, and we don't have 2 cohorts, we have 4. All right? So bear with me, this is all going to be a little weird. First off, t-test is intended for continuous data. Counts are not continuous. Fair enough? I mean, I had 3 spectra identified for this protein, or I had 10. But I didn't have 3.2, right? That makes count data not continuous, and t-test expects continuous data. So I'm instead going to use what's called a Poisson model. And Poisson models, I mean, they have the drawbacks too, but at the very least, they can be used in spectral count data. But I have 2 factors at work. So I think a lot of people know about some of the crop proteomics work that take place in the department. This is kind of a modification of a model I made for somebody a couple years ago. So here I have a generalized linear model. Does everyone remember linear modeling? Okay, so we could all write down our ages and our masses. And you would see that us senior people have been working on our mass quite well, right? So there's a correlation between the two. So we could use a linear model to ask how robust is this relationship between our ages and our excellent masses. And a linear model is how we would do that. But a linear model can do lots of interesting things. In this case, we have a linear model that's using 2 factors. I have samples that differ by salinity, low salt or high salt, and by moisture, not enough water, plenty of water. So I want to ask, how much do these two factors play into the spectral counts that differentiate our 4 cohorts? So I might have a control that's low salinity, low moisture, or normal moisture, and I have one that's high salinity, low moisture. I have the low salinity and high salinity, high moisture as well. So you see I have 4 boxes essentially. And these boxes are all in a square. So I want to compare these versus these, and I want to compare these versus those. Okay, so I've now created a linear model where my spectral counts are expected to be a function of both salinity and moisture using a Poisson family of statistics. Poisson is a type of statistical model appropriate for count data. Okay, now I can run an Anova, an analysis of variance. Anyone heard of Anova? Okay, sometimes you don't have just 2 cohorts, sometimes you've got more. Anova is something that handles the more case. So I make an Anova of this model, I use a chi-square test with it, and then from that I can grab out p-values that are associated with what's the probability associated with salinity being differential or moisture being differential. This is how you would do it in R. There are lots of languages you could do this stuff in, but this is just one of them. Okay, so in many cases, dealing with count data becomes rather more problematic because they are not continuously distributed. You can learn about Croissant regression from this little blue link at the left. And you can see a fine example in the case of spectral count data from affinity purification experiments by Choi et al. Back in 2008. It's another case where they used Poisson modeling to analyze spectral count data. Okay, so that's a bit of math, and now I'm going to just move on to the next slide while I'm still ahead. No one fell asleep right then. That's really good. Well done, everybody. All right, volcano plots. Everybody loves a volcano plot, except the statisticians. Statisticians do not love volcano plots, but you should know how to read one. Because very, very frequently, when you have differential expression data from transcriptomics or from proteomics, volcano plots are where we do that assessment. You also see them in stuff like metabolomics quite frequently. So what we have is a plot in which we have a fold change, here called enrichment, on log scale. Okay, so when I see a value of 0 on a log scale, I remember that means 1 to 1. So this means way up in controls, this means way up in cases, let's say. If it's in the middle, that means there's no fold change between the two. So if anything that falls in this central column of the figure, essentially it's not a very big fold change. When we use this, we're putting some limits on things to say, we don't care about fold changes less than this. And this is the part that statisticians actually hate. They hate it very, very much. But biologists frequently tell us, we don't care if it's up by 10%. That's not biologically meaningful. That's what these 2 lines mean. The next part is that when we do our statistical test, some things had very low p-values and some things had very high p-values. We really want to emphasize those things that had very low p-values. That's why we flip this all inside out and use a negative log of the p-value. That means very low p-values are very high on the volcano plot. And p-values that are 0.5 or whatever are down here at the bottom. Okay? So this most popular part of the volcano plot, the bottom center section, is the set of proteins that neither had a good p-value nor did they have a big fold change. This is the land of lost toys here, right? This is the I don't know, the stuffed animal that you have stuffed in your closet somewhere and really should have gone into a dumpster many, many years ago. Right. These are proteins at the bottom left and bottom right that did have big fold changes, but they were not significant because they had high p-values. Thus, they're low on this plot. So the really good stuff is in the upper left and upper right hextents of this model, because those are proteins that had a big fold change in one direction or in the other, and they're high enough to indicate that they had very low p-values. So low p-value, high fold change, this is the biologist's dream. And in this case, the software created by Goodhart and Leisterberg? How would you pronounce that? It's got an IJ in it. Americans aren't equipped for that.
Leisterberg.
What?
Leisterberg.
Whoa! Leisterberg.
It's Listenberg. Listen is English.
Oh.
Leister, listen.
Ah,
No, that's Dutch.
that's Dutch. Okay. All right. So a very interesting paper. They put forward some software that you can use to make your very own volcano plots. It's pretty cool stuff. So the proteins here are up in cases. These are proteins that are up in controls. This is a useful way to separate it out. These show up everywhere, so it's good to know about them. Now, I cannot give a lecture on this topic without discussing multiple testing correction because it's really important. Before, I showed you a spectral count table that had 500 or 600 rows in it, and each of them had count. We had 500 and 600 rows of these. So what this means is that we're going to have to produce a test for each and every one of those proteins. So each row is associated with a particular test. It's just the same as it would be in the microarray scenario. Here, however, we actually have continuously distributed data. We could use Ttest. We don't have to do all that generalized linear model stuff here. But still, we need multiple testing correction because each row has its very own test. So why do we need it? Because if you do a bunch of T-tests or a bunch of linear model tests, however you're doing it, we expect that 1 in 20 of these tests will give us a significant p-value even when no difference exists between the cohorts. That may not seem sensible, but it's true. If you simply work through a big table of random numbers that are generated from exactly the same distributions, you will find that you detect differences by random chance 1 in 20 times if you're using a p-value of 0.05 as your threshold. That's because the p-values are distributed uniformly across the 0 to 1 range. So that's a problem. So if you perform 1,000 T-tests, 1,000 proteins, 1,000 genes, whatever you're doing, 1,000 metabolites for that matter, you should expect that 50 of them will be significant, which is to say less than 0.05, by random chance alone. It has nothing to do with biology, everything to do with random variation. Okay, so there are 2 primary ways that you see people using multiple testing correction. The first of these is the Bonferroni correction. Bonferroni correction is really, really tough correction. If you want to see all of your false hits go away, Bonferroni is great for this. But it's quite likely to kill the proteins you really thought were truly differential, because Bonferroni is extremely punitive. The role of Bonferroni is to protect you from making any false positive claims. Benjamini-Hochberg, or FDR correction, is intended to limit the rate of false claims. The rate of false claims. So, if you're going to use this strategy, and everyone must use multiple testing correction when you're doing even 100 T-tests, let alone 1,000, it's much more common to see people using the Benjamini-Hochberg or FDR approach to say, of the differences I'm claiming, we expect that fewer than 5% of them are false positives. Okay? So there are lots of tools out there that can do this sort of multiple testing correction for you. It's simply something that we must always take into account. When you're looking at a volcano plot, it's worth knowing whether they've done multiple testing correction on these or not, because sometimes they're not. Okay, so we move to our final section's takeaways. There are plenty of things that we learned from microarrays, things like volcano plots and MA plots and things like that have simply been repurposed for use in proteomics. Sometimes the way that we build our data in proteomics, such as using spectral counts, can be very problematic when we try to simply apply the same lessons from microarray statistics in proteomics. So be aware of that distinction. If you're going to become an expert biomedical scientist in the area of proteomics or bioinformatics or transcriptomics, you should really assume that you're also going to need to learn a lot about biostatistics to prevent yourself from making mistakes along the way. So having all the biostatistical training you can get is really not a bad idea. I know sometimes it feels like it closes our eyes. Biostatistics is actually a really interesting subject if you start looking into some of the interesting characters who created it. All right, so label-free quantitation has become a widespread technique for differential and quantitative proteomics. I'm kind of hedging myself to ask about what's different is a little different than saying by how much. So I'm kind of teasing those apart by calling it differential and quantitative proteomics. Spectral counting and extracted ion chromatograms start from exactly the same data. The same experiments can be interpreted both ways. But the way that we interpret those data, the way that we build protein ratios out of them are completely different. So it's really worth understanding which of those two you're working with. Continuous and discrete quantities, which is to say values like XICs tend to be continuous, values like spectral counts are discrete. They require different kinds of statistical models. You shouldn't really just assume that I did Ttest here, so I should do Ttest there. It's really important to use the right kind of test for the right kind of data. Okay, and that's the summary for label-free quantitation, and I hope that I have not confused you worse, but rather made this a little bit more accessible. Because at some point in your life, you're going to be doing some sort of cohort versus cohort comparison. Great, thank you.